Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Sufi Huzaimi bin Muhammad Fauzi. Our topic is about Krebs cycle and electron transport cycle. What is Krebs cycle and what is the history behind this cycle? Krebs cycle is introduced by Sir Hans Adolf Kreb in 1937, and another name for this cycle is TCA cycle, which means tricarboxylic acid cycle. This cycle is a series of enzyme catalyst reaction that can decrease the acetyl-CoA and inner compartment of mitochondria called mitochondrial matrix. Next, the Krebs cycle is the primary metabolic pathway for all the aerobic processes in an animal tissue by which carbo, fats, and amino acids are oxidized to form carbon dioxide and water. In Krebs cycle for aerobic organism, it is an amphibolic pathway, ones that participate both in the catabolic and anabolic processes. Now is electron transport cycle. The product of Krebs cycle will enter the electron transport cycle to continue the process in producing a lot of ATP. This cycle, also known as respiratory chain, which resides in intermembrane space of mitochondria. This ETC cycle contains 4 multi-subunit mitochondrial complexes that help in the transporting electrons coupled with the translocation of protons across the inner mitochondrial membrane. This generates an electrochemical gradient or proton motive force across the membrane, which derives the conversion of ADP to ATP.
Pyruvate derived from glycolysis is oxidatively decarboxylated to acetyl-CoA by the pyruvate dehydrogenase in order to enter the Krebs cycle. Now, I will explain about the Krebs cycle. There are 8 steps in Krebs cycle. Firstly is the formation of citric acid. The first step is a condensation step, combining the 2-carbon acetyl group from acetyl-CoA with a 4-carbon oxalic acetate molecule to form a 6-carbon molecule of citrate. The enzyme used is citrate synthase. Second step is the formation of isocitrate. Citrate loses one water molecule and gains another as citrate is converted into an isomer isocitrate by isonitase. Third step is the formation of alpha-ketoglutarate. The third step is the formation of alpha-ketoglutarate. This reaction is catalyzed by the enzyme isocitrate dehydrogenase. First, isocitrate is dehydrogenase to form oxazosinate. It undergoes spontaneous decarboxylation to form alpha-ketoglutarate. The NADH generated in this step is later oxidized in electron transport chain (ETC) to generate ATPs. Isocitrate with 6 carbon undergoes oxidative decarboxylation to form alpha-ketoglutarate. Next, for the fourth step is the formation of succinyl-CoA. Alpha-ketoglutarate is oxidatively decarboxylated to form succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase. The NADH thus generated enters into ETC to generate ATPs. Another molecule of CO2 is removed in this step. The fifth step is the generation of succinate. The next reaction involves a substrate level phosphorylation whereby a high energy phosphate is generated from the energy trapped in the Chester bond of succinyl-CoA. The enzyme used is succinate thiokinase. The sixth step is the formation of fumarate. Succinate is dehydrogenated to fumarate and unsaturated decarboxylic acid by succinate dehydrogenase. The hydrogen atoms are accepted by FAD. The FADH2 then enters into ETC to generate ATPs. The succinate dehydrogenase is competitively inhibited by malonites. The seventh step is the formation of malate. The formation of malate from fumarate is catalyzed by fumarase. The reaction involves the addition of water molecules. The eighth step is the regeneration of oxalic acetate. Finally, malate is oxidized to oxalic acetate by malate dehydrogenase. The coenzyme is NAD+. The NADH is generated in this step, which enters the electron transport chain when ATP is produced. To be summarized, the product which are NADH and FADH is used in the electron transport chain.
Bismillahirrahmanirrahim. Assalamualaikum. My name is Muhammad Akmal and I will explain about electron transport chain mechanism in this part. Electron transport chain is the last part of aerobic respiration, which the only part that uses atmospheric oxygen. And in this part, there are 4 complexes of protein that are labeled from 1 to 4, numeric Roman style. And please take note that at the end of the process, the electrons will reduce the molecule of oxygen and produces water. In complex 1, an ADH will release 2 electrons to flavin mononucleotide that is located in the complex 1 protein, in the protein complex, and the electron is then passes to iron sulfur, which will then be transferred to coenzyme Q in the second complex. In complex 1, 4 hydrogen ions will be transferred across the membrane from matrix of mitochondria to intermembrane space. At complex 2, coenzyme Q will combine with 2 electrons from matrix of mitochondria and get converted to reduced form of ubiquinone. In complex 2, an enzyme will reduce the hydrogen in succinate to produce fumarate and generate FADH2 that is located in the complex, and it will then transfer electron to iron sulfur and transfer it to ubiquinone. The ubiquinone will then transfer the electron received from complex 1 and complex 2 to complex 3. In the third complex, it consists of cytochrome B, cytochrome C, and also iron sulfur. Cytochrome is a prosthetic group of heme which is similar to hemoglobin, but it carries electron and not oxygen. This results the iron ion to reduce and oxidize as it passes electron to cytochrome B. The electron received from cytochrome B, which will then be transferred to cytochrome C to be transported to the fourth complex. And please take note that in this complex, in the third complex, 4 hydrogen ion is being pumped to intermembrane space. In the fourth complex, it composed of cytochrome proteins C, A, and A3. This complex have 2 heme group and 3 copper ions. The cytochrome will hold an oxygen tightly until the oxygen is reduced, and the oxygen will then pick up 2 hydrogen ion from the surrounding to make water. And in this complex, 2 hydrogen ion will be passed across the membrane from the matrix of mitochondria to intermembrane space. From the transport chain, it showed an uneven level of concentration between matrix of mitochondria and intermembrane space. The hydrogen ion could not pass through the membrane without the help of an enzyme. Hence, ATP synthase will act as an enzyme that will convert ADP to ATP by receiving hydrogen ion from the intermembrane space, which is a high concentration, and pass through the membrane into the matrix of mitochondria, which is lower concentration. And this can conclude that the conversion of ADP to ATP will generate energy. So that is all from me. Thank you.
Hello and good day to all of you. I will be summarizing this video, which is on the topic of Kreb cycle and electron transport cycle. Okay, on to the first topic, which is the Kreb cycle. Okay, for the Kreb cycle, it is a process to produce the necessary molecule for the energy transport change, for example, NADH, FADH, or GTP. And also being used in metabolism process to break down glucose, lipid, and amino acid to their monomer form. And if the Kreb cycle does not occur in the body, first thing first that will happen is the cell body cannot be repaired or renewed because there's no energy. And then the second thing that will be happen that the food that we eat cannot be break down, which will cause further health issue as for example, constipation. And then we go for the next topic, which is the electron transport cycle. The first thing that we need to know about this cycle is that it is the main source of the ATP production. And also, the electron that's being used is used to pump the hydrogen ion into the intermembrane space to form water or to be used again for the ATP production. And what if the electron transport cycle does not occur? Well, there will be no movement of hydrogen ion, which means there will be no ATP production, thus no energy will be created. So when energy cannot be created, weakness will occur throughout the body, as there is no energy, and the deterioration of muscle and tissue as the cell use the energy from the muscle and tissue to produce more energy. And that's all from me. Thank you.